Hi everyone, I'm Jack Killalea. I'm a PhD candidate at the University of Queensland, and I'll be presenting my honours thesis study that was completed last year. Firstly, thank you to my supervisors, Professor Leanne Hydes and Dr. Zoe Walter, and the Lives of Well Research Group at UQ for their guidance and contribution to the current study. And thanks to APSAD for having me here. Cannabis continues to be liberalised across parts of the world, though our understanding of which individuals and what contexts are most vulnerable to its adverse effects remains elusive. The robust association found in numerous longitudinal and experimental studies between cannabis and psychotic-like experiences, or PLEs, is one example. As little attention has been devoted to investigating this relationship and potential underlying mechanisms within the context of daily life. Therefore, the current study aimed to examine the cannabis and PLE relationship on the daily level and assess whether day-to-day fluctuations in negative affect and facets of self-regulation moderate the relationship. A daily diary approach was utilised to capture the past day experiences of a community sample of 89 young adult cannabis users with varying degrees of recent PLEs over 10 days. Improving on previous diary studies, a standardised joint unit was used to quantify cannabis use. This was calculated by adding the number of self-reported doses together, with each dose type weighted relative to a standard cannabis unit, and multiplying this by the proportion of cannabis to tobacco reported. This variable highly correlated with self-reported grams. PLEs were measured by the 15-item CAPE measure, negative affect was probed by 2 items, and facets of self-regulation were measured by the 12-item momentary self-regulation questionnaire. Multi-level modelling of the daily data indicated that daily cannabis use predicted PLEs, as did days marked by heightened levels of negative affect and emotion dysregulation, relative to each participant's mean level across the study period. Furthermore, only daily levels of emotion dysregulation moderated the daily cannabis and PLE association, seen in the figure here. These results remain significant after adjusting for other daily substance use and a number of baseline covariates. To conclude, this is one of the first diary studies to test and identify potential psychological mechanisms underlying PLE vulnerability associated with cannabis use. Heightened levels of emotion dysregulation were found to strengthen the daily relationship between cannabis use and PLEs. The study's findings highlight the importance day-to-day fluctuations in affective processes have on exacerbating PLEs in the context of cannabis use. Though future research is needed to properly examine the directionality of these variables, given the large 24-hour window of assessment. Thank you.